Welcome to Eukaryotic gene regulation. This is kind of like the part 2 of our of yesterday's lectures, so hopefully you'll enjoy. Instead of talking about the lac operon, we're going to talk about the yeast Galfor system, which is needed for the metabolism of galactose in yeast. Now, this is hopefully obvious to many of you, but in case you have forgotten, yeast are not bacteria. E. coli is a bacterium. Bacteria, including E. coli, are prokaryotes. Meaning they have sort of circular chromosomes and no nucleus. On the other hand, yeast are eukaryotes, just like us. Meaning, they are a lot larger than bacteria, and they have a nucleus where they store their DNA. The gDNA in there. This is the nucleus. These are our happy budding yeast cells. All right, hopefully that makes sense. Here, we'll color them in that color. And then E. coli, I always like to make pink. Let's give them a face too. All right, happy E. coli, happy yeast, completely different types of organisms. All right, moving right along to the gene regulation portion of our video today. So I told you we're going to do the Galfor system. And it's already moving around on me. So yeast, among other proteins that are involved, yeast possess 4 key genes needed for the uptake and metabolism of galactose so it can be used as an energy source. No, I don't. I want to do that later. We installed that before. My computer distracting me. The names of these genes are GAL1, GAL2, and then you guessed it, it's not going to go in order, GAL7 and GAL10. Why would it need to go in order? Biologists are naming these things. And I'm going to pause for one moment. All right. And the key feature of this suite of genes is we want transcription to be turned off when galactose is unavailable. You don't need to metabolize galactose if there is no galactose to metabolize. It's a waste of energy. However, when galactose is present, you want to turn these genes on so you can actually use it as an energy source. Sort of like lactose in the lac operon situation. So in order to achieve these goals, yeast cells expressed 3 regulatory proteins, called Gal4, Gal80, and Gal3. Oh, I don't know where the numbers are between, Gal10 and Gal80, but that's just not how they were named. I've given up on spelling protein correctly. Gal4 is, of course, the superstar in this regulatory network, but everybody is important. So first, let's talk about Gal4. Gal4 is actually the only transcription factor in the bunch because it's the only one that can bind DNA. And it is a transcriptional activator, a really powerful one. So I had a lot of fun developing a little character for Gal4 that reflects its functional domains. So let's have at it. I drew it as like a couple of little cucumbers pooching out with some DNA binding domains. Here's the DNA. And then, here's their beautiful little goofy Gal4 faces. That one turned out creepier than I intended. Okay. And so Gal4, its DNA binding domains are zinc fingers. So different in the lac operon, we were dealing with exclusively homeodomain DNA binding domains. Gal4 uses zinc fingers. You may notice it is also a dimer. So two Gal4s get together to form a functional activator. Each dimer has 2 zinc fingers, one after the other. So a total of 4. There is also, as you may imagine, a dimerization domain that allows the 2 Gal4 subunits to connect. And there is an activation domain that allows Gal4 to do its job. So one of those protein-protein interaction domains. Now I want to color Gal4 in. So in green, we have sort of its activation domains and its dimerization domains kind of both slammed together. And here in, I think yellow, we'll have our zinc finger DNA binding domains. And just for funsies, here's the DNA that Gal4 actually is bound to. Gal4's preferred binding site is called UAS. UAS stands for, helpfully it's meaningful, is upstream activation sequence. Because it always resides upstream to the promoter. So here's the core promoter of any gene, really. Oh, why are you doing this? Okay. And then here would be the actual gene coding region, and UAS is upstream of all of that. So that's why it's an upstream activation sequence. Gal4's preferred binding site is actually pretty simple. It is CGG. And because it's binding as a dimer, it's going to be one of those repetitive binding sites with the inverted repeats. So on the other hand, it should be CCG. This would be the plus strand. And on the minus strand, shown from 3' to 5', you have CGG and then CCG. In between those C's and G's should be 11 nucleotides. Gal4 is pretty flexible about what those nucleotides actually are, but it needs to have 11 of them for the dimer to be able to fit on the UAS. So this is a total of 17 nucleotides that make up the Gal4 consensus sequence. And can also be named UAS, has many names. And then once again, because Gal4 is binding as a dimer, you'll notice that 5' to 3' on one end of the UAS is CGG. On the opposite strand, starting at the 5' end, is also CGG. The complementary strands likewise are inverted repeats. So you have CCG here and CCG here. So this already indicates to you that Gal4 must bind at least as a dimer or a tetramer. We know now that it's a dimer. So Gal4 likes to bind UAS, and that's good because UAS sequences can be found upstream of all of the Gal genes. So Gal4 has the power to turn on all of the other galactose metabolism genes 1, 2, 7, and 10. All right, so that is Gal4. Now I want to tell you about Gal80. Gal80 is a Gal4 repressor. It is not a transcription factor because it has no DNA binding domain. However, it does indirectly regulate transcription, so it's a little confusing. Transcription factors must bind DNA. Well, let's see what it looks like. So to see what Gal80 looks like, we had to first draw Gal4. That is one of my lesser attractive straight lines that I try to draw sometimes. All right. So here is UAS. Here is happy Gal4. And what Gal80 does is a dimer of Gal80 hides Gal4's activation domain. And due to this, we're going to draw out a really creepy face. And now they look like little twin mushrooms. Isn't it beautiful? In the video game of life. So I'm going to color everybody in. We'll do Gal80 in a nice repressive red color. Gal4 should be green because it is an activator. And its DNA binding domains are yellow because that's the color of zinc fingers in my mind. And here's the UAS DNA. And so what happens is Gal80 binds to and masks the activation domains of Gal4. So they cannot activate. So when Gal80 is present, Gal4 can still bind DNA. That's not the problem. However, its activating ability is gone. Because Gal80 is hiding its strong activation domains like a weird little mushroom cap. So Gal80, we have Gal4, our activator, Gal80, our repressor of Gal4, and now we need a repressor of Gal80 or something to make this cycle complete. And hence comes Gal3. So Gal3, we'll draw as this little guy, is a binder of galactose and Gal80. So it actually can bind Gal80 when it is bound to galactose. So here is Gal3. Here is a nice Gal80 mushroom cap. And when you add galactose, Gal3 is able to bind it. And then it is able to bind Gal80 very tightly. Gal80 is along for the ride. Let's color everybody in. All right, so galactose, we're just gonna make it pink because why not? Gal3 is gonna be purple. And Gal80 is our nice red color. So in quick review, yeast want to be able to turn on the Gal genes, Gal1, 2, 7, and 10, when galactose is present, so they can break it down and use it for energy. These genes should come on when there's galactose and turn off when galactose is not there. So we use our 3 regulatory proteins, activator Gal4, repressor of Gal4, Gal80, and capturer of Gal80, Gal3. Gal4 is a dimer that binds DNA with zinc finger DNA binding domains. It has a strong activation domain that allows it to start transcription at the genes where a UAS site exists. When Gal80 is around, it binds to Gal4 and disguises its activation domain so they don't work anymore. When Gal3 is around, it can bind galactose and then bind to Gal80. And in the next video, we'll see the consequences of this interaction here. So I will tune back in for part 2. See you soon.